In mathematics and physics, vectors are used to represent quantities. Vectors have magnitude and directions. In general, vector is something which is used to transfer some product or stuff from one place to another. In biology, a vector does the same work. In molecular biology, vector is a DNA molecule that is used as a vehicle to carry the foreign genetic material artificially into another cell so that it can be expressed or replicated. Vector is a molecule that is used as a vehicle to transport foreign genetic materials artificially into another cell so that it can be replicated and expressed. A vector that contains a foreign DNA is called a recombinant DNA. There are four major types of vectors. They are viral vectors, plasmid vectors, cosmids and yeast artificial chromosomes. Origin of replication, a selectable marker and a multi-cloning site lies in common for all engineered vectors. The vector is normally a DNA sequence itself. It contains an insert and a large sequence that functions as a backbone of the vector. The intention of a vector is to isolate, multiply and express the insert in a target cell. Expression vectors are vectors that specifically express the transgene in the target cell and normally have a promoter sequence to drive expression of the transgene. The transcription vectors in a target cell can replicate but not express, unlike expression vectors. To amplify their insert, transcription vectors are used. In bacterial cells, the insertion of a vector into a target cell is normally called transformation, and in eukaryotes it is called transfection, although insertion of viral vector is called transduction. Plasmids are the vectors that are most commonly used. They are double stranded DNA molecules that have the capability of automatically replicating in a host cell. Plasmid vectors consist of an origin of replication. So, plasmids are self-replicating in the host cell and also transgene insert. Bacterial plasmids contains special genes resistant to antibiotics. More features like multiple cloning site are generally present in modern plasmids. The multiple cloning sites include nucleotide overhangs for insertion of an insert and multiple restriction enzyme consensus area to either side of the insert. When plasmids utilized as transcription vectors, incubation of bacteria with plasmids will generate hundreds or thousands of copies of vectors in the bacteria within few hours. Further, vectors can be extracted from the bacteria, and using the restriction enzymes, multiple cloning sites can be cut to remove the 100-fold or 1000-fold amplified insert. In a characteristic manner, this plasmid transcription lacks crucial sequences that code for translation termination sequences and polyadenylation sequences in the translated mRNAs. This makes the protein expression from the transcription vectors impossible. The plasmids may be transmissible or conjugative and non-conjugative. Transmissible or conjugative mediates the DNA transfer through conjugation and thus spread rapidly among bacterial cells of a population. For example, F plasmids, many R plasmids and few Col plasmids. A plasmid that cannot proceed to another bacterium during conjugation but needs mediation by a conjugative plasmid is called as non-conjugative plasmids. In other words, the plasmid will not affect conjugation and self transfers to another. Viral vectors are vectors that are genetically engineered to carry the modified viral DNA or RNA that is being depicted non-infectious but still contain the transgene and viral promoters, further allowing for the translation of the transgene through a viral promoter. For large scale transfection, the viral vectors need helper viruses or packaging lines since they lack infectious sequences. Viral vectors are designed for permanent incorporation of insert into the host genome, and thus genetic markers are left in the host genome after incorporating into the transgene. A necessary component in all vectors is transcription. Even stable expression is thus determined by a stable transcription that normally depends on the promoters in the vector. Expression vectors are constitutive or inducible. This expression is based on various promoter activities and not the post transcriptional activities. Thus, the two different expression vectors depend on different types of promoters. In bacteria, the plasmids are found as small circular double stranded DNA molecules. They are sometimes present in eukaryotic and archaea organisms. Plasmids carry genes that may benefit the survival of the organism and frequently transmit from one bacterium to another through horizontal gene transfer. In molecular cloning, artificial plasmids are widely used that serves to drive the replication of the recombinant DNA sequences within the host organisms. The size of the plasmid varies from 1 to more than 1000 kilobase pairs. The identical plasmids number in a single cell may range from 1 to 1000 under some conditions. Johann Lederberg first introduced the term plasmid in the year 1952. Plasmids are considered as replicants since it has the ability to replicate autonomously with a suitable particular host. In all the three major domains, plasmids can be found. The domains are archaea, bacteria and eukarya. Host to host plasmid transfer needs direct mechanical transfer by conjugation or changes in host gene expression that allows the uptake of the genetic element by transformation. The gene that is to be replicated is inserted into a plasmid that contains genes and makes the cells resistant to specific antibiotics and a multiple cloning site that is a short region containing many commonly used restriction sites that allow the easy insertion of DNA fragments at this location. Next, by transformation process, the plasmids are inserted into bacteria. The bacteria are then exposed to the specific antibiotics. The bacteria that take up copies of the plasmid only survive, as the plasmid makes them to be resistant. In detail, the protecting genes are expressed and the protein that is expressed breaks down the antibiotics. By this way, to select only the modified bacteria, the antibiotic acts as a filter. Now, these modified bacteria can be grown in large amounts, harvested and cut to isolate the plasmid that is of interest. Large amounts of proteins can be made using plasmids, which is another main use of it. Researchers grow bacteria that contain a plasmid maintaining the gene of interest. To confer its antibiotic resistance, the bacterium produces proteins and it can also be induced to synthesize large amounts of proteins from the inserted gene. To mass produce a gene or protein, this is a cheap and easy way. A phage or phasmid contains an F1 origin of replication from an F1 phage. When combined with the filamentous phage M13, it can be used as a type of cloning vector. A phage can be replicated same like a plasmid and also be packaged as a single stranded DNA in the viral particles. For double stranded replication, phage contains an origin of replication called ORI region as well as an F1 ORI to modify a single stranded replication and packaging into the phage particles. Many generally used plasmids contain an F1 ORI region and thus are phagemids. Similar to a plasmid, a phagemid can be used to clone the DNA fragments and introduce into a bacterial host by many techniques like the electroporation and transformation. Infection of the bacterial host that contains the phagemid with a helper phage, for example M13K07 or VCS M13, gives the necessary viral components to enable the single stranded DNA packaging and replication of the phagemid DNA into the phage particles. The phage particles are secreted through the cell wall and released into the cell cytoplasm. The filamentous phages normally retard the bacterial growth, but the lambda phage and the T7 phage is not generally lytic. The helper phages are generally engineered to package less efficiently than the phagemid. So, the resultant phage particles contain predominantly the phagemid DNA. The F1 filamentous phage infection needs the presence of a pilus, so only the bacterial host containing the F plasmid or its derivatives can be used to produce phage particles. Phagemids were used to generate the single stranded DNA template for sequencing purposes. Nowadays, phagemids are also useful in producing the templates for site-directed mutagenesis. The detailed characterization of a filamentous phage life cycle and the structural features lead to the development of phage display technology, where a range of peptides and proteins can be expressed as fusions to the phage coat proteins and displayed on the viral surface. Within the phage particle, they displayed polypeptides and peptides are associated with a corresponding coding DNA. So, this technique contributes itself to the study of the protein-protein interactions and other ligand or receptor combinations. A cosmid is a type of hybrid plasmid often used as a cloning vector that consists of a lambda phage cos sequence. Hence, cosmids can be explained as cos site plus plasmid gives rise to cosmid. DNA sequences of cosmids are originally from the lambda phage. To build the genomic libraries, cosmids can be used. With the bacterial ORI-V, cosmids are predominantly plasmids, and ORI-V, V stands for vector, has an antibiotic selection marker and a cloning site, and they carry one or more cos sites derived from the bacteriophage lambda. Depending on the specific intention of the experimental, broad host range of cosmids, shuttle cosmids or mammalian cosmids are available. Depending on the size of the vector, the loading capacity of cosmids varies. The cloning procedure involves the generation of the two vector arms which are then further joined to the foreign DNA. The selection against wild type cosmid DNA is simply done through size exclusion. Cosmids always form colonies and not the plaques. Through a technique called in vitro packaging, the total DNA is transferred into an appropriate Escherichia coli host after the construction of recombinant lambda or cosmid libraries. The essential packaging extracts are derived from Escherichia coli C1857 lysogene. These extracts will identify and package the recombinant molecules in vitro, yielding either mature phage particles or recombinant plasmids that are present in the phage shells. These differences are then reflected in the different infection frequencies seen in favor of the lambda replacement vectors. This will compensate their slightly lower loading capacity. Phage libraries are also stored and screened easier. The genomic DNA has to be cut into the suitable size range of restriction fragments and cloned. This is normally done by the partial restriction followed by either dephosphorylation or size fractionation to avoid the chromosome scrambling, that is the ligation of the physically unlinked fragments. A bacterial artificial chromosome, simply called as BAC, is a DNA construct which is based on a functional fertility plasmid, that is the F plasmid, and is used for cloning and transforming in bacteria, especially in E. coli. F plasmids play an important role as they contain partition genes that elevate the even distribution of plasmids after bacterial cell division. The usual insert size of the bacterial artificial chromosome is 150 to 350 kilobase pairs. From the bacterial P1 plasmid, a similar cloning vector called PAC has also been produced. In genome projects, BACs are generally used to sequence the genome of organisms. Example, the Human Genome Project utilizes BACs. In BACs, a short piece of the DNA of an organism is amplified as an insert and then it is sequenced. Then finally, the sequenced parts are rearranged in silico, which will result in the genomic sequence of the organism. In modeling the genetic diseases, BACs are now being used widely, often alongside the transgenic mice. When studying neurological diseases such as Alzheimer's disease or aneuploidy that is associated with the Down syndrome, BACs have been used to some degree of success with the mice. BACs were also studied for specific oncogenes that are associated with the cancers. They are transferred by transfection, electroporation or transformation with a suitable virus or microinjection to these genetic disease models. Using BAC arrays, BACs can be used to detect genes or large sequences that are of interest and then use it to map them onto the human chromosome. As BACs accommodate much larger sequences without the risk of rearrangement, they are preferred for these kinds of genetic studies. BACs are also more stable than other types of cloning vectors. The genomes of many large DNA viruses and RNA viruses have been cloned as BACs. They are referred as infectious clones, as the transfection of the BAC construct into the host cells is sufficient to initiate the viral infection. BAC's infectious properties have made the study of many viruses like the herpes viruses, coronaviruses and the pox viruses accessible. Using genetic approaches, molecular studies of these viruses can now be achieved. Such genetic approaches depend on either linear or circular targeting vectors to carry out the homologous recombination. Yeast artificial chromosomes are chromosomes that are engineered genetically and derived from the DNA of the yeast Saccharomyces cerevisiae, which is further cut into a bacterial plasmid. The inserted sequences can be cloned and physically mapped using a process called chromosome walking by inserting large fragments of DNA, that is from 100 to 1000 kilobase pairs. For the Human Genome Project, this process was initially used. Due to stability issues, yeast artificial chromosomes were abandoned from the usage of bacterial artificial chromosomes. By discovering the essential autonomously replicating sequences, simply called as ARS in the initial research, the inherently fragile chromosome was stabilized. The centromere, ARS and telomeres from Saccharomyces cerevisiae are the primary components of an YAC. In addition to these components, selectable marker genes such as antibiotic resistance and a visible marker are being utilized to select the transformed yeast cells. During the extracellular replication, the chromosome will not be stable without these sequences and will not be distinct from colonies without the vector. A YAC is constructed using an initial circular DNA plasmid. Using the restriction enzyme, the initial circular DNA plasmid is typically cut into a linear DNA molecule. To ligate the DNA sequence, ligase is used, which forms a single large circular piece of DNA. The basic linear yeast artificial chromosomes can be broken down by 6 main steps. They are ligation of the selective marker into a plasmid vector, differential selection of the colonies with or without the marker gene is being allowed. The presence of LUU2 gene allows the YAC vector to get amplified and selected for an E. coli by helping the mutant E. coli to produce leucine in the presence of the components within the growth medium. A mutation in the yeast host cell induces the accumulation of the red pigment. Insertion inactivation of the gene is caused by cloning a foreign DNA fragment into the YAC and restores the red color. Thus, the colonies that consist of foreign DNA fragment are red in color. Ligation of essential centromeric sequences for the mitotic stability, ligation of autonomously replicating sequences called ARS that provides an origin of replication to undergo the mitotic replication, which in further allows the plasmid to replicate and multiply extra chromosomally, but delivers the plasmid highly mitotically unstable, and without the centromeric sequences it is easily lost. The yeast expression vectors such as yeast integrating plasmids, YIPs, and yeast episomal plasmids, YEPs, have an advantage over the bacterial artificial chromosomes BACs, where they can be used to express the eukaryotic proteins that require the post-translational modification. Since YAC has the capability to hold a large fragments of DNA, it can be utilized to clone and assemble the whole genomes of an organism. The YAC can be propagated as linear artificial chromosomes by the insertion of a YAC into yeast cells, and cloning can be done by inserting the regions of DNA in the process. Two processes can be used to obtain a sequenced genome or the region of interest after this process. The processes are physical mapping and chromosome walking. In this session, we discussed about the vector elements of the DNA molecule, the role of plasmids, phagmids and cosmids. We also discussed about bacterial artificial chromosomes, that is nothing but BAC, and yeast artificial chromosome, the YAC. Vector is a molecule that is used as a vehicle to transport foreign genetic materials into another cell so that it can be replicated and expressed. A phagmid or plasmid contains an F1 origin of replication from an F1 phage. When combined with the filamentous phage M13, it can be used as a type of cloning vector. A cosmid is a type of hybrid plasmid often used as a cloning vector. Cosmids can be explained as cos sites plus plasmids is equal to cosmid. The bacterial artificial chromosomes, that is BAC, is a DNA construct which is based on a functional fertility plasmid F plasmid and is used for cloning and transforming in the bacteria, especially Escherichia coli. The yeast artificial chromosomes, that is the YAC, are chromosomes that are engineered genetically and derived from the DNA of the yeast Saccharomyces cerevisiae. Hope all of us are knowledgeable enough with this topic on vectors. It will help you to further your studies on DNA, which ultimately helps in genetic transformation.